Hello, my name is David Abramson, and on behalf of the staff and the International Board of Trustees of the Israel Cancer Research Fund, I would like to welcome you to the 2022 ICRF City of Hope Jacki and Bruce Barron Cancer Research Scholar Program Symposium, titled New Targets and Therapies in Cancer.
And I'm Beryl Chernoff. The Jacki and Bruce Barron Cancer Research Scholars Program Symposium is a culmination of a 5-year collaborative effort between the Israel Cancer Research Fund and City of Hope Hospital. This collaboration was the great vision of Harvey Miller, through which many of City of Hope's top scientists were partnered with the best and brightest scientific cancer researchers in Israel.
With generous support and underwriting from the Harvey Miller Family Foundation, Jacki and Bruce Barron, and the Norman and Sadie Lee Foundation, 16 separate world-class research projects were granted. Today, you will hear some highlights of the most recent efforts.
We would also like to recognize Dr. Nancy Mizels, Chair of ICRF's International Scientific Council, who will serve as our moderator today. Special thanks also go out to ICRF team members Ellen Rubin, Caitlin Schmidheiser, and Allie Marks Greenfield for all their help in making today possible. We would also like to thank Advanced Concepts, who are providing all the technological magic for today's event.
As this is the last symposium of the initial 5-year project, let's now listen for just a few minutes to some of the goals, dreams, and hopes of this amazing collaborative effort, best told by those who made it possible.
I don't think cancer discriminates. Cancer impacts everyone's life at some point or another. If it's not you, it's someone in your family, it's a close friend, it's an acquaintance. I can't overestimate the importance of cancer research and the need for a cure.
Jacki and I got involved with ICRF in 2010. Steve Rosen, Dr. Steve Rosen, was treating Jacki, and he came to us during actually one of Jacki's treatments and told us that he was being honored by ICRF and asked if we'd ever heard about the organization.
We looked into the organization. It looked like they did wonderful work. And at that point, I was so sick and he was saving my life. So I don't think there's too many things he could have asked us to do that we would have said no to.
So when Bruce called me and asked me if I wanted to be the dinner chairman, my immediate thought was yes. And then I started looking for ways of honoring that commitment. I called Dr. Rosen. I gave him the idea that I had, which was this collaboration effort between City of Hope scientists and Israeli scientists, hoping that these great minds could work together and come up with medication, come up with treatment, come up with more than the cancer world has seen before.
We had no idea that at the gala that Harvey was going to surprise us with this amazing gift of a collaboration.
It's not just the immediate collaborations that they wrote the grants for. If you were at any of the symposiums, one doctor from one collaboration would go up to another doctor from another collaboration and say, well, you did this and this and this with these cells, and could that be applied to something totally this or that? And they would start talking, and it was just amazing to me, the relationships that were going to develop and the incredible science that was going to come from it.
We had 12 projects. They were broad in spectrum. They ranged from the genetics of breast cancer to defining a new target for the treatment of acute leukemia, to understanding the mechanism of drug resistance in lung cancer related to the microbiome that involves bacteria growing in the lung. And in addition, a better understanding of how the immune system controls the growth of cancer and how we can take advantage of the immune system to kill cancer. We are so appreciative of all the support and the efforts of our donors and our scientists. The friendships that were developed and the camaraderie is beyond description, and we anticipate will lead to many more important initiatives.
You know, I think that $5 million is a lot of money, and 5 years is a long time. And I think that in the world of drug discovery and drug development, you know, $5 million is not a lot of money, and 5 years is probably not a lot of time. But the question that Harvey and I have discussed with Jacki, the three of us have discussed, is, so a foundation has been laid, but what's next? This is a template. There are other institutions that can get together and follow Harvey's vision and leadership to form similar types of collaborations.
Cancer research must be funded. The cancer research can't stop. When something like cancer hits close to home, it becomes a much more important item than just an article in the paper. And Bruce and Jacki are close to home. In fact, I don't know if Bruce remembers this. My wife used to play mahjong with his mother many years ago. I guess the whole purpose of the program and the whole purpose of the research is try and make sure that it doesn't hit too many homes. So for people out there wondering why this video, why all the effort, the more we can put into the discussion, the more we can put into the research, the faster we will reach the conclusion. And the conclusion to me is the elimination of cancer.
Welcome. I'm Nancy Maisels. I'm chair of the International Scientific Council of ICRF, and I'm really pleased to be chairing or moderating this virtual symposium, the 2022 Barron Scholars Research Symposium. And the title of this symposium is New Targets and Therapies in Cancer. The generous gift from Harvey Miller is supporting teams of collaborators at City of Hope and at different schools in Israel, schools and research institutions. And you'll hear from one member of each of a variety of teams, and they'll be talking about different kinds of cancers for which they've developed treatment: breast cancer, melanoma, lung cancer, T-cell lymphoma, myeloma, pancreatic cancer. And they'll be talking about a range of different targets and therapies, from immunotherapy that's being done by training one's own cells to go after the tumor, to therapies targeted to bacteria, which have been shown to grow within different tumors. So between those two extremes, there's lots and lots to hear about. All very new, all very exciting. So I'm delighted to welcome you to our exciting program, and I hope you enjoy hearing about the science as much as I. There'll be no time at the end for questions because this is a virtual recording, but we do welcome questions, so make note of your questions. The program will have email addresses of the speakers, and they would all welcome questions from the audience. So here we go. So our next speaker will be Ariel Munitz, who will be talking to us about how eosinophils, a specialized cell of the immune system, can be trained to attack tumors. And Ariel is usually at Tel Aviv University, but he'll be speaking to us tonight from the shores of the Dead Sea, where he's preparing to run a half marathon tomorrow morning. Ariel.
Yes, thank you very much for this kind invitation to speak and the even more generous introduction. And I'll be talking to you today about the project which we have been in collaboration with Who Are You from the City of Hope regarding the roles of eosinophils in metastasis, and specifically in breast cancer metastasis. So Eosinophils are usually cells that have been studied in context of allergic inflammation, and they are a major drug target in inflammatory diseases that are related to allergy, such as asthma. However, over the last couple of years, new roles which have been exceeding far and beyond allergy have been discovered for these cells. For example, eosinophils have key roles in obesity and in metabolism. Eosinophils have multiple roles in liver and muscle regeneration, so they can actually promote tissue repair. And even new innate and adaptive immune responses have been now attributed to eosinophils. So it is quite known now that eosinophils can participate in settings which are far and above and beyond allergy. But one thing that we don't know is we don't know too much about eosinophils and the tumor microenvironment. It is an area which is entirely understudied, and this is quite interesting because we know that eosinophils can accumulate in tumors for more than 100 years ago. So what was our approach? What we decided to do is, first of all, we were asking, what are the roles of eosinophils in breast cancer-driven metastases? And the reason for that is we know that the lung is an area which recruits multiple metastatic cells, and we know that eosinophils are present and active in the lung from settings of allergy. And the first thing that we wanted to answer was, are eosinophils present in human metastases? So we obtained lung metastases of breast cancer patients. You can see here a patient on the left and a patient on the right. And you can see these brown dots and sometimes even brown smear, which indicates the presence of eosinophils. This is a granule protein, which is a protein that eosinophils have within them. And when the eosinophil gets activated, it can just pour out its content. And you can see that on the right, you have patient number 18848, which has very massive deposition of this granule protein which eosinophil possesses. And on the left, patient 178532, you can see that that patient has rather eosinophils which are not that activated. So we don't exactly know how and what activates eosinophils in human disease. And therefore, we went ahead to study what happens with eosinophils in mouse models or experimental models of lung metastasis, which are breast cancer-driven lung metastasis. And what we have been able to show pretty nicely, and sorry, this is quantification of the previous slides that I show, that about half of the patients are degranulating eosinophils, which means active eosinophils, and the other half are non-degranulating. But having that in mind, we really wanted to know what eosinophils do in this tumor microenvironment. So going into experimental settings, we went and established mouse models of breast cancer-driven lung metastasis. And what we could nicely show is that eosinophils are very rapidly recruited into the tumor microenvironment, and this is initiated by tumor-secreted factors. So within a given tumor mass, there are now eosinophils which are resident in the lungs, and there are eosinophils which have been recruited from the peripheral blood. These cells, both recruited and resident eosinophils, are exposed to lymphocytes which are present in that tumor microenvironment, such as CD4 positive T cells and NK cells. And these cells provide cytokines. These are soluble proteins which can activate these eosinophils. So eosinophils which are present in the tumor microenvironment are activated by these soluble factors. And what eosinophils do is they secrete another protein which is called chemokines. And these chemokines now attract CD8 positive and CD4 positive T cells, which can now kill the tumor. So basically what we have shown in this very nice collaboration with HUA is that eosinophils are present and are active in lung metastases. Eosinophils display anti-tumorigenic activities, and this is done by interacting with T cells. Eosinophils and T-cell interactions are, we think, a very important axis in terms of anti-tumorigenic activities, specifically in lung metastasis, but also in additional tumors as well. And I would like to finish off by thanking, obviously these findings have significant, what we think, implications in targeted cancer immune therapy. And I would like to finish by thanking, first of all of our collaborators on this work. As you can see, it has been an international effort, and of course the ICRF for funding this research.
Thank you, Ariel. That's really exciting. So you've gotten the eosinophils to act on your behalf to recruit other cells. They're kind of intermediaries. in fighting cancer. What happens after you've stimulated the cells in this way and the immunotherapy is complete? Do the eosinophils continue to be involved?
So what we can show, and this is ongoing studies that we're doing right now, is just like you mentioned, when we activate these eosinophils, we take the exact, I would say, proteins which we know to induce these anti-tumorigenic programs in eosinophils. We stimulate them and we give them back to a mouse which has tumors. We can actually see a very nice reduction in tumor mass. And this reduction in tumor mass is accordingly also with increased or elevated levels of CD4 and CD8 positive T cells, indicating that we can really reprogram eosinophils to recruit T cells. And this has implications in cancer immune therapy because the eosinophils in the tumor microenvironment can persist for a very long time. They do not go away. So once we stimulate them, they are there and they can continue to recruit T cells. And one thing that is quite interesting, and this is something that we see from clinical observations, there's a very nice correlation, positive correlation between patients that have been treated with targeted T cell cancer immune therapy, such as anti-CTLA4, anti-PD-L1, or anti-PD-1. And if they develop eosinophilia, they have actually a better prognosis. So there seems to be some interaction between T cells and eosinophils also in the clinical setting. We don't exactly know what this interaction is, but this is definitely something that we're working on right now.
Right. And it's clear that you're poised to be able to enhance this interaction in people, in human cancer patients, and that it is very likely going to have some sort of use in the clinic. That's really wonderful. Thank you so much for telling us about it.
Thank you for this opportunity.
Thank you for taking the time the night before the half marathon. I want to wish you good luck on behalf of everybody at ICRF. We'll be watching your performance.
Thank you very much.
Tomorrow. Yeah.
Thank you. I'm planning to break the record.
Good, good.
Toda.
I'm delighted to introduce Ravid Straussman from the Weizmann Institute. His lab has shown us that there are bacteria living in tumors and contributing to their response to therapy. And he's now going to tell us how the microbiome, all the little creatures that live in a tumor, affect the response of lung cancer to treatment. Ravid.
Thank you very much. It's a pleasure to be here. So this is a joint project that I did with Dr. Dan Raz from City of Hope. And the idea was really to characterize for the first time the bacteria that are present inside human lung cancer and to also understand their effects on response to therapy. So lung cancer, as you probably know, is the highest death toll as compared to all other cancer types. And before studying the effect of the bacteria there, we wanted to understand which bacteria are present inside lung cancer. And we published this paper together at 2020 in Science, demonstrating that in the majority of lung cancers and also other cancer types, one can find bacteria. We demonstrated that the lung tumors have a unique microbiome and that there is some specific bacteria that are present in lung tumors, but not in normal lung. We found the bacteria to be present both inside the cancer cells and in immune cells. And we found that in tumors of smokers, there are many more bacteria than in tumors of non-smokers, and a unique bacteria. We also found that bacteria in tumors from smokers are enriched in specific functions that can help them deal with it. For example, we found in these bacteria in enzymes that can degrade nicotine. Now, to understand what these bacteria can do, we started with an in vitro screen. So we took lung cancer cell lines with different mutations. We treated them with drugs, different drugs, with or without 60 different species, bacterial species. What we found was that in 2 cases of lung cancer cells that had the EGFR mutation and were treated with EGFR inhibitor, or had the ALK translocation and were treated with ALK inhibitor, we found specific bacteria, not all of the bacteria, but very specific bacteria that can really rescue the cancer cells from these targeted therapies. So here's an example. This is a control. When we have cancer cells in green, you can see them. When you add on bacteria, there is no much effect. You have a drug and you kill the cancer cells. But when you add the drug together with a specific bacteria, you rescue the cancer cells. Now, a few years ago, I had a paper in which I demonstrated that bacteria sometime in pancreatic cancer can degrade the drug, and that's how they confer resistance to chemotherapy. So both Dr. Raz and myself, we followed these two stories in our lab trying to understand the mechanism. How can bacteria rescue cancer cells from chemotherapy? And what we found, without going into much detail right now, for the sake of time, is that unlike in the pancreatic cancer story, when bacteria degraded the drug, here the bacteria secreted specific metabolites that can activate unique receptors on the cancer cells and can bypass the block. So to give you an example, let's say this is a receptor, an EGF receptor or ALK receptor, and usually when these are mutated or if there is translocation, they signal to the cancer cell to proliferate all the time. But when you block it, you usually kill your cancer cells, but when you have bacteria, they can bypass the block and reactivate the proliferation of the cells. And we think that these discoveries are revealing novel drug targets that can help to overcome drug resistance if we understand them better and target them. So I'd like to thank again for the funding that we got for this, and hopefully this will be published soon. Thank you.
So thank you, Ravid. One of the questions that comes immediately to my mind as a biologist is whether there are other organisms in addition to bacteria, microorganisms, also living in tumors, viruses, fungi. Do you know anything more about this?
Yeah, so that's a great question. We didn't look much on viruses, but we do have another work about fungi in which we characterize the same tumors for fungi. And indeed, we found that in many of these tumors, there are fungi. More than that, we found that these fungi sometimes correlate with specific bacteria inside the tumor. So there's like couples of specific bacteria and specific fungi that like to live together. And we also are following these interactions and their effect on cancer immunity. It's a really interesting field.
And do you think these fungi might also contribute to resistance to chemotherapy and might therefore be targets to enable chemotherapy to work better?
Yeah, so we don't have enough evidence there. What we did see, and this is unpublished yet, is that these fungi, as well as the bacteria, can really affect the immune landscape of the tumor. So one can speculate that they will affect the way patients respond to immunotherapy, but this is something that is an ongoing work in the lab.
This is just tremendously exciting and brings us real hope for new avenues of therapy in tumors that have been resistant to very standard treatments. So I'm very, very grateful to you for doing this research and for sharing it with everybody. Thank you so much.
Absolutely. It's our pleasure. Thank you.
I'm delighted to introduce our next speaker, Christiane Querfeld, who will be telling us about new approaches to treating T-cell lymphoma. Christiane.
Thank you. Thank you for the opportunity to speak and to show our exciting data on reprogramming of tumor-associated macrophages with anti-PD ligand 1 and lenalidomide in cutaneous T-cell lymphoma. So what are the issues and what are the problems to solve in cutaneous T-cell lymphomas? So what we know is that M2-like tumor-associated macrophages are the most abundant phenotype that promote the growth of cutaneous T-cell lymphoma, hereby inducing immunosuppression. Little is known, however, about the underlying mechanisms that induce a programmed cell death 1 or PD-1 positive a PD-1 positive M2-like tumor-associated macrophage phenotype in cutaneous T-cell lymphoma. So we identified PD-1 positive M2-like tumor-associated macrophages that accumulate in the lesional skin of cutaneous T-cell lymphoma. And now the problem to solve is, how can we identify drugs that will reprogram these tumor-promoting PD-1 M2 tumor-associated macrophages to tumor-counteracting M1 macrophages? So our experimental approach and results are seen here in this slide. And our comprehensive analysis of patient blood and skin tissue samples from lesions of cutaneous T-cell lymphomas revealed critical cytokines, gene signatures, and signaling pathways that are involved in the development of PD-1 positive and M2 tumor-associated macrophages. And we were also able to induce this in an in vitro model, and hereby using cutaneous T-cell lymphoma cell lines and monocytes from blood samples. So we identified the same pathways involved in the M2 polarization that we have seen in blood and skin tissue samples. So here's a sketch. What you can see, this is the tissue culture. We got this M2 tumor-associated macrophages. Now, how can we reprogram them, however, to M1 macrophages? So we found that anti-PD-ligand-1, or durvalumab, and the immunomodulatory agent lenalidomide could reprogram the tumor-promoting PD-1 positive M2 macrophages into M1 macrophages, and they were able to attack the tumor cells and help to strengthen an immune response against cutaneous T-cell lymphoma. So what you see here is on the slide is on the top here is macrophages that can now finally, the M1 can phagocytose cancerous cells. We also found that those M1 macrophages, you can see here, was applied to these drugs, can actually downregulate immune suppressive cytokines and have upregulation of pro-inflammatory cytokines that help to reprogram the whole microenvironment. So the impact on the cancer is M2-like tumor-associated macrophages are abundant immune cells within the tumor microenvironment and associated with tumor growth and progression. So the tumor microenvironment, which makes up of immune cells that are surrounding the cancerous cells, is in a dynamic state, and targeting those tumor-associated macrophages polarizing is a novel therapeutic strategy that is not only relevant for cutaneous T-cell lymphoma, but also for many other hematologic and solid malignancies.
So thank you, Christiane. That's very exciting. So to recap, what you've done is take a cell type that would normally stimulate tumor growth, and you've converted that cell type to a cell that's fighting the tumor, as we hope our immune system will do. And that seems like it might have impact, not, as you said, not just on T-cell lymphoma, but other tumors. How do you see this impacting directly onto patient treatment in the near future?
I think the impact of this treatment will enhance all treatments we have so far. In cutaneous lymphomas, treatments are established, don't induce long-lasting responses. So now, but these treatments just target the cancerous cells. Now we target the microenvironment, meaning like we target immune cells that support the tumor growth to reprogram them to learning to attack the tumor cells again. And so this will enhance the responses. So not targeting only cancerous cells, but the whole environment, the whole ecosystem, so to speak, that nourishes those tumor cells. And so this is very exciting.
Yeah, no, it's tremendously exciting. Thank you so much for sharing that with us. And we look forward to learning more results in the future.
Thank you. And thank you for the opportunity.
It's my pleasure now to introduce Idit Shachar. She's going to be telling us about a new therapeutic target in multiple myeloma named CD84. Idit.
Hi. So I'm telling you today about multiple myeloma. Multiple myeloma is a malignancy caused by expansion of malignant antibody-producing cells in the bone marrow. The over median survival of the patients is 5 to 6 years. And the stages are, we can see stages of differentiation of the disease. And the microenvironment in this disease plays an important role in the tumor progression. So our focus is on a SLAM family that has several members in this family, and our study concentrate on one member of this family, which is called CD84 or SLAMF5. This member of CD84 is a member of the SLAM family, which all function in the immunological synapse. So the unique of the SLAM family is that they don't have ligands. There is a homophilic engagement, cell-cell contact, that triggers the redistribution of freely diffusing SLAMs into the immunological synapse, and this leads to downstream signaling events. So it looks like this. If we have two cell types like B cell and T cell, and the two express CD84, so this is the homophilic engagement. This engagement leads to cell type-induced signaling events that result in specific outcomes. Each cell has its specific outcomes. So previously we have shown that CD84 serves as a survival receptor in chronic lymphocytic leukemia. We showed that interaction of CD84 expressed on the malignant cells with the CD84 expressed on the microenvironment leads to cell survival. And our question was, what is the role of CD84 in multiple myeloma? So in order to answer this question, we first looked at malignant cells, multiple myeloma cells derived from patients, from bone marrow human patients. And you can see that expression of CD84 is elevated during the progress of the disease, suggesting that CD84 has a role in multiple myeloma. But in order to understand what is the role of CD84, we had to move to a mouse in vivo model. And we used the model in which we inject malignant cells into mice, wait 14 days, and after 14 days, we start to inject CD84 blocking antibody, which is called B4, or a control antibody. And then on day 21, we analyze the different organs. So what we saw is that when we block CD84 in the multiple myeloma model, we see that the mice less die. It means that the blocking CD84 inhibits the death of the mice, and also it inhibits the paralyzation of the mice, suggesting that CD84 plays an important role in multiple myeloma. We also looked at the tumor load, and we saw that there is a strong downregulation in the tumor load in the injected mice. You can see the spleens are different. These are the SLAMF5 injected mice, and these are the control mice. And we also looked at the exhaustion markers on T cells. The T cells are very important in the immune response, and we saw less expression of tumor exhaustion markers and elevated activity of the T cells. So we then wanted to see what happens, in human cells. So we took multiple myeloma cells from human and incubated them with the antibody, the CD84 blocking antibody. And we can see that blocking CD84 decreased the survival of the malignant cells and reduced the cell numbers, decreased the T cell exhaustion, as we saw in our mouse in vivo model, so we see less exhaustion markers. And together we suggest that it has a similar role in the human cells compared to the mouse murine cells. So overall, we suggest that stroma cells and cells in the microenvironment express CD84. This CD84 mediates the interaction with the microenvironment. It regulates the expression of the exhaustion markers on the T cells, and that's why T cells do not respond. When we block CD84 with our antibody, we see that T cells are more functional, and these T cells can kill the malignant cells.
So thank you, Edith. So just to summarize, so people who aren't familiar with the science are sure to understand that the tumor, multiple myeloma, lives in an environment of its own, really. It's like a little ecosystem with different kinds of cells trying to respond, trying to destroy it. And you found one of the key molecules that regulates that response. So hopefully there will be an antibody available for people with multiple myeloma that's effective just like the antibody is in mice. That's great. I'm pleased to introduce our next speaker, Zwoming Sun from the City of Hope, and he will tell us about a new approach to therapy of T-cell lymphoma. Zwoming?
Thank you. First of all, thanks Jacki and the Bruce Barron Cancer Research Scholars Program for supporting this collaborative research between Dr. Noah Asokov from Israel and me, Zwoming Sun from City of Hope. So our question is, what is the relationship between cancer cell and the immune system? At the earliest time, the relationship between cancer and the immune system are mostly ignored. So the research focus either on immune cell or cancer cell individually. Until recently, the Nobel Prize winning discovery showed that immune cell actually can reject cancer. However, our research found that when you stimulate immune response, particularly this Th17 immune response, it can lead to cancer growth. So here is the mouse with a gene mutation in the TCF1. This is before induction of Th17 response. This is after induction of Th17 response. You can see this lymphoma develop in the thymus. This is a developing T cell. Therefore, our result shows that immune response can promote cancer growth. So our question becomes, what is the function of TCF1 in Th17 immunity and the lymphoma development? Dr. Asokov's lab has the expertise in understanding signaling molecule function in T cell and the lymphoma. So his research focused on the cancer cell. My lab has the expertise understanding immune response, so my research focus on understanding the TCF1 function in the immune cell. The result from both of the lab shows that TCF17 immune response produce cytokines to stimulate lymphoma proliferation. What happened is in the normal cell, I mean in the wild type, the gene locus for IL-17 and the IL-2 are closed. So the normal function of TCF1, you can consider it as a lock. It keeps those gene locations closed or locked. However, when TCF1 is mutated, this lock mechanism is disrupted. So allow the gene location for IL-17 and IL-22 opened. That lead to gene expression and the overproduction of IL-17 and IL-22, which then stimulate lymphoma cell proliferation. However, those cytokines don't just stimulate any cell proliferation. they only stimulate lymphoma cell precursor cell proliferation. Why? Dr. Asakov's study shows that PCOT1, this gene, is a positive regulator for cell proliferation. It's upregulated in lymphoma cell. On the other hand, CCND2 is a negative regulator for cell proliferation. It's downregulated. And the more PCOT1 upregulation is responsible for the downregulation of CCND2. So this gene expression change make lymphoma cell susceptible to stimulation-induced proliferation. And again, I try to show you Dr. Asakov's study is on this part, and the MATLAB is on this part. So what is the implication of our research on cancer? So the current popular immunotherapy, such as use a checkpoint inhibitor, is to stimulate immune response that allow immune cell to reject cancer cells. Our study shows that this stimulated immune response could lead to cancer growth instead of a cancer rejection. And because our study is in the mouse, so the question is, what is the relation to the human lymphoma? Actually, TCF mutation was found in human lymphoma, in this type of human lymphoma that has only 19% survival rate. And our study indicate that enhanced Th17 response and upregulate the PCOT and the CCND2 in the lymphoma cell could be responsible for this lymphoma, and therefore targeting Th17 response and the PCOT and the CCND2 could be the potential therapy for this kind of human lymphoma.
Thank you very much. That was really interesting, Zhoming. There's an obvious question that comes to mind, of course. If a patient were making a decision about whether or not to have immunotherapy, how would they weigh your result? Does it contraindicate immunotherapy, or does it mean the immunotherapy would have to be modulated so as not to cause the tumor to grow?
Yeah, that's a very good question. So whenever we have the immunotherapy, we have to, keep this in mind. Although we meant to reject cancer, our research indicated it could lead to cancer growth. So we have to check whether those cancer is due to the mutation in TCF1, whether this patient has mutation in TCF1. That's one. So more research is required to understand under what circumstance it lead to cancer growth, under what circumstance lead to cancer rejection. So we need more understanding that process, and more understanding to separate the immune response that reject cancer growth and cancer rejection and the cancer growth, so we can separate this immune response specifically target to cancer rejection.
So that makes sense, and that's a really interesting result, and I hope that you're able to learn more about this because it sounds like it could be quite important. Thank you very much.
Thank you.
It's my pleasure now to introduce Tomer Shlomi, who will be telling us about metabolic engineering of T cells as a way to improve immunotherapy. Tomer.
Thank you very much. I'm happy to present our research together with Professor Saul Preisman from City of Hope, funded by ICRF on overcoming immunosuppression in solid tumors through metabolic engineering of T-cell therapy. Cancer immunotherapy is a promising approach for fighting tumors, and it is based on harnessing the immune system to fight cancer cells. It is very commonly applied these days for hematological tumors and it is less effective against solid tumors, and especially against common solid tumors such as breast cancer, colon cancer, prostate cancers, and others. Much of the research on cancer immunotherapy focused on unleashing immune cells from signaling suppression. This is achieved using, for example, immune checkpoint inhibitors and so on. Another source of immune suppression that takes place in the tumor microenvironment is metabolic suppression. So it is not enough to trigger immune cells to fight cancers. They also need to have metabolic nutrients in the tumor microenvironment to be able to survive, proliferate, and exert their activity against cancer cells. This has been a major focus of cancer metabolic research in the recent 5 years or so, which identified numerous metabolites whose concentration goes down in the tumor microenvironment and enables cancer cells to evade the immune system. For example, cancer cells deplete glucose and glutamine from the tumor microenvironment and starve T cells, preventing them from surviving and actively killing cancer cells. Cancer cells also secrete some metabolites that inhibit the activity of T cells in the tumor microenvironment. Our goal is to overcome this metabolic immunosuppression in the tumor microenvironment. We focus on using cell-based immunotherapy and specifically CAR T therapy. So in CAR T therapy, a patient blood is taken, and specifically T cells are isolated. These T cells are engineered and grown ex vivo before being infused back to the patient. What we are doing is we are adding another step in this process, which involves metabolic engineering. We are trying to rewire the metabolism of T cells such that they would not depend on the availability of some specific metabolic nutrients in the tumor microenvironment. So basically, we want to change the metabolism and boost it in a way that they would work better in terms of surviving and proliferation in the tumor microenvironment. So how do we do that? Well, we developed some genetic screening approach, which enables us to identify metabolic genes. These are genes that code for metabolic enzymes, whose overexpression in T cells provides them with metabolic benefit. So how do we do that? So we start from a library of metabolic enzyme coding genes, and each of these genes are associated with a DNA barcode. We take T cells and transduce them with a pooled library of such metabolic genes. We then perform adoptive cell therapy in a tumor-bearing mice using these T cells that were transduced with metabolic enzymes. We extract a tumor, and this tumor would have different populations of T cells penetrating it, whose survival depend on the overexpression of the different metabolic genes. So to assess the abundance of the different metabolic of the different T cell subpopulations in the tumor, we perform next generation sequencing, which enables us to identify specific T cell subpopulations overexpressing specific metabolic genes. So we apply this approach both in Technion and in City of Hope, using different CAR T therapy models, one of them for melanoma and another one for prostate cancer CAR T therapy. And this is a model that was developed originally in City of Hope. The goal here is to identify new metabolic enzyme coding genes whose overexpression in T cells improve the efficacy of CAR T therapy, enabling T cells to overcome metabolic limitations in the tumor microenvironment. Now, being able to identify such metabolic genes would improve our ability to apply CAR T therapy not only for hematological cancers, but also for solid tumors. Thank you very much.
So Tomer, that's very exciting. People have been really waiting for this solid tumors to be effectively attacked by CAR T cell therapy. Can you tell us if you have discovered any candidates that might be useful?
We've identified several candidate enzyme coding genes even before completing this genetic screen based on applying mass spectrometry metabolomics analysis of a tumor interstitial fluid. So we take tumors, we analyze the fluid inside the tumor, we search for metabolites whose concentration go down there, and based on that, we can rationally design which metabolic enzymes with overexpression would be beneficial for T cells. So we have several candidates for that, and we're looking forward to have additional candidates from this genetic screen.
Excellent. So you're really well positioned to solve this very difficult problem. Thank you so much for doing this research and for sharing it with all of us, and lots of success in the future. Thank you. I'm delighted to introduce our next speaker, Ziv Gill, who will talk to us about treating pancreatic cancer by targeting pyrimidine metabolism pathways. Ziv.
Thank you very much. I'm going to talk about a significant problem in cancer treatment, about patients with pancreatic cancer. In the upper slide, you can see that among 9 different cancers, you can appreciate that we've done quite good research and improvement in treating patients with cancer within the last 4 decades. You can see that from the 70s till the 21st century, there's improvement in most of the cases of cancer, but there's one disease that the outcome and the improvement of survival during the last 40 years was only 1%. And this is patients which have pancreatic adenocarcinoma. One of the main reasons for this small improvement in the outcome of patients during the 40 years is that pancreatic cancer have significant resistance to all known chemotherapies. And this includes one of the most common drug in oncology, which is gemcitabine. Gemcitabine is a common drug, a chemotherapy that it was given to pancreatic cancer patients and to other patients with cancer. And you can see in the lower figure that this drug enters the cell and undergo different changing in its form, change in its formation before it entering the nucleus of the cells and creating nicks and breaks in the DNA of the pancreatic cancer cell. One of the reasons for the resistance of pancreatic cancer to this chemotherapy is a group of molecules known as purine and pyrimidines. You can see here, this is represented in the right lower side of the figure, GTP, ATP. All these molecules are increased de novo, they are increased by the pancreatic cancer cells, and that is a main cause of resistance against chemotherapies and specifically against pancreatic patients treated with gemcitabine. Now, the group at City of Hope, Lale Mellstrom's group, came with a very interesting and genuine idea, is to take a drug which is approved by the FDA, which is used for treatment of other non-cancerous disease like rheumatoid arthritis, and this drug is known as leflunomide. And they thought and speculated that maybe with this drug we can improve the effect of gemcitabine as chemotherapy. Now, in the same time, across the Atlantic, my group in Haifa were working on very unique group of cells. You can see in the upper left panel cells which are stained in green, and they're moving very fast, creating close interaction with the gray cells, which are pancreatic cancer cells. Now, what we found is that these cells, which are immune cells known to be as tumor-associated macrophages, they secrete very small bubbles, vesicles in the size of 80 nanometer. And inside these bubbles, there are a lot of goodies. There are proteins, RNA molecules, and also metabolites, including these pyrimidines and purines that increase the resistance of pancreatic cancer cells against gemcitabine. In the lower slide, in the lower panel, you can see the pancreatic cancer cell with the small green bubbles, which came from these immune cells, from these tumor-associated macrophages. These vesicles enter the cancer cells and increase the level of the purine and pyrimidines, increasing resistance to gemcitabine. And in the upper left panel, you can see that whereas gemcitabine alone was a very poor treatment against pancreatic cancer in mice, when they combined that with the drug leflunomide, the size of the tumor shrinks significantly, almost by more than half. Now, the fact that we can treat now successfully as a proof of concept against mice with the drug, an FDA-approved drug with a very low cost, and you can see here that you can buy in Costco for $30, you can buy this drug. Not only the study that provides proof of concept for treating patients with pancreatic cancer, but also it can provide cancer treatment at very low cost, reducing the cost of treatment of pancreatic cancer patients that can reach $16,000 per month. So this drug will be also available for low-income countries that will be helping the treatment of pancreatic cancer in these countries, as well as in the United States and in Israel. Thank you very much.
Well, thank you, Ziv. This is wonderful work. Thank you for doing it. And it's tremendously exciting that you've taken an existing drug, put it to new use, and that you can envision it's going into the clinic and being successful in the near future. Can you tell us a little more about the next steps and how long they might take?
So our study provides a proof of concept on mice that this drug can be of significant value for cancer treatment. And because this drug has very low toxicity and is actually approved for treatment of other diseases by the FDA, it can reach very fast for for entering a phase 1 treatment in pancreatic cancer patient, and that needs relatively 100 patients is enough, and then going to step 2 and phase 2 and phase 3 relatively fast, because the need for improvement of outcome in this patient is significant.
So this is great. Thank you very, very much. It's my pleasure to introduce Erez Levanon from Bar Ilan University in Israel, who will tell us about a new kind of target for therapy of breast cancer and other cancer. Erez.
Thank you. I want to describe our projects that we did together with Kevin Morris from City of Hope. Now he's moved for Australia, so it's not working in City of Hope anymore. And this project was done together with Shai Ben Aroya and Eli Eisenberg from Israel. So we are speaking about RNA mutation, and we know that typically people, when speaking about mutation in the cancer, they speak about DNA mutation. So let's think about it for a minute. Why DNA mutation is so important for cancer? And this is very simple, because mutation in the genome eventually will lead for alteration of a protein that key for the progression of the cancer. But because the process of developing of a mutation is random, for each mutation that is critical for the cancer progression, the cancer genome accumulates a large number of mutations that are not relevant for the cancer, are simply passenger mutations for the progression of the cancer. But if you look at the flow of information in the cell, you notice that information is not only located in the genome, but also in the RNA. And we know that the RNA can also have alteration in the content, but they're considered to be much less important, simply because a variation at the RNA will not going to retain for the next generation of cancer cells. Therefore, even if there is something that's beneficial for the cancer, they will not retain for the next generation of cancer cells, and therefore there will be no selection in favor for this type of variation. Yet, we know that variation at the RNA is very common, and we see them across many cancer types. Specifically, in breast cancer, we find a high numbers of variation at the RNA. And we call this variation RNA editing. And the question that we want to ask is simply why? Why so many cancers have large number of variation in the RNA seemingly have no advantage for the cancer? In order to do so, we first develop a computational approach to allow us to find the variation of in the RNA. We call them RNA editing. And utilizing this approach, we have to say that it's much more difficult to find variation in the RNA than the DNA, because variation in the DNA, it takes place in the all molecule of the DNA in the same cell. We simply have 2 copies of the genome in the cells. But for each RNA molecule, we have many times hundreds or even much more copies of the same gene, of the RNA of the same gene, but only few of them undergo variation. So it's much more demanding computational to find this variation. So we developed computational approach to find this variation. And by that, we first found, for the first time, the global atlas of where variation of 18 take place in the human genome, where this event consistently take place in any genome that in any healthy genome. But now, and this is most exciting part, and this experiment done in the Kevin Morris labs, we found experimentally that when we took cancer cells, that we can increase the editing activity. We elevated the expression level of the protein that's responsible for this editing, this protein called ADAR. We see that they can overcome and have much higher tolerance for chemotherapy. So this means that when you have high level of editing, the cells, it's much easier for them to overcome chemotherapy. And this opened a completely new and very exciting idea, that while you have mutation in the DNA, it's in the long run, it's very important for the cancer cells, because to accumulate for the key event in the critical for the cancer progression. However, you pay the price to have mutation all over, which many of them are very difficult, very dangerous for the cell. But when you have high number of variation in the RNA, you don't have to pay this price, because after a few days, all the RNAs vanish with all the variation. So you can allow yourself, the cancer, to have a large number of a huge diversity, in the hope that one of these variation can help them to overcome the resistance and become resistant for chemotherapy. And this means a practical meaning and implication. You can consider develop new drug against ADAR, the editing activity, and to think about how to treat with chemotherapy new cancer patient. And I want to thank you very much for the support in this project.
So Erez, thank you very much. This is really exciting, because it seems that what you've told us is that a protein normally expressed by cells, ADAR, that edits RNA, is expressed at higher levels in cancer, causes resistance to chemotherapy. And if you can target that protein, cells will no longer be resistant to chemotherapy and they'll no longer evolve more quickly. Is that correct? That's enormous.
This is a big hope. I'm sure there is many additional mechanisms. Cancer is so complicated, but it seems that this is completely overlooked mechanism so far. And this, the unique about of this mechanism that it's completely take place in a different arena of the cell, not in the DNA, but rather in the RNA. And I hope and I believe that is open a way and a door for completely new world of a new understanding of how specifically drug resistance mechanism can overcome.
This is just wonderful. I really want to thank you for doing this work and for sharing it with us. It really gives many people new hope for how their cancers can be treated. So on behalf of ICRF and everybody in the world, I thank you for your work and for your presentation.
Thank you very much.
I'm delighted now to introduce John Termini, who will be telling us about some wonderful results from a long-standing collaboration between him at City of Hope and Ziv Gross at the Technion in Israel. John.
Thank you, Nancy. It's a pleasure to be able to present some of our work here at the Jacki and Bruce Barron Cancer Scholars Program Symposium. I've been working with Professor Gross for a number of years, and it's been a wonderful collaboration and very fruitful. And we've had a lot of fun doing work together and hopefully developing some very useful therapeutics which would benefit people with cancer. What we're working on is a type of molecule called metallocarols. And there's a new concept in cancer therapy called theranostic, which combines the ability to image a tumor and then with the same molecule, with the same modality, to then provide therapy to kill that tumor in a single type of frame. And this, we call this theranostics. And one of the molecules that we've been exploring for a number of years to perform these kinds of functions are called metallocarols. And they provide a framework or scaffold for developing these kinds of drugs. You can see on the bottom, there's a structure that looks kind of like a crown. And in the middle of that crown, there's a metal. In this case, it's gallium. And these metallocarols have a very highly fluorescent, and they're tunable. And they're tunable in the sense that by taking that metal, in this case, gallium, and replacing it with something like silver or gold or manganese or iron, we can change not only the tumor imaging properties, but also its cytotoxicity and its ability to kill tumor cells. And the Gross lab at Technion has really been a pioneer in the synthesis and application of these molecules. We've seen selective uptake in cancer cells, and I'll mention and discuss a little bit about how they can be modified with targeting proteins to improve their activity. So on this slide, you see an example of our uptake. We've looked at a number of different kinds of cancer cells. These are just cells now on the left panel. And this is an example with melanoma cells, and you could see the red panel on the top. The second box shows the uptake of metallocarols into these melanoma cells, and it's highly selective, and you can see the red fluorescence is very intense. And if you look right next to it in the blue, that's blue is indicating the nuclei of these melanoma cells. And if you go over to one more where it says merge, I hope you can see that the red and the blue are co-localized in the same place, which means that these metallocarols in melanoma cells are specific for targeting the nucleus, which is the source of their cytotoxicity. Now, when we do that in an animal model, we see some different things. So on the right, we have a mouse model of breast cancer, and you could see the two tumors on the right and left flank of the mammary pads. And when we inject metallocarol into the tail vein of that mouse, you could see that the imaging on the slide, the picture box on the left, kind of goes everywhere. It's not very specific. But if we then combine that with a protein called heregulin, which targets HER2-positive breast cancer cells, I hope you can see that the action is now very specific and the imaging is very tightly regulated about the two tumor sites. So this combining with proteins really is helping us improve the specificity and targeting a number of different kinds of cancer cells. So in some, metallocarols combined with these targeting proteins are promising theranostic drugs. We've shown applications to melanoma, breast, and recently we've been looking at prostate cancers using a different type of protein targeting strategy to both image and then kill prostate cancer cells, again in an animal model. So a recent modification of this technology, we've looked at the ability to enhance their lifetime in serum, circulating lifetime, by combining them with nanoparticles, and this seems to extend their lifetime in vivo and allows them to stay longer to target their tumor cell targets. So in closing, I'd like to give many thanks to the ICRF, the Barron Family Foundation, and Harvey Miller for their generous support of our work over the years. Thank you for your attention. I'd be happy to answer any questions.
Well, thank you, John. And this is really wonderful. It's sort of a now you see it, now you don't. You can visualize the tumor cell and also kill it in one swoop, combining these two kinds of reagents, the metallocorol and the proteins. The obvious question is, what do you foresee as the timeline for developing this as a therapy for treatment of, say, malignant melanoma or breast cancer?
Yeah, that's a great question. Thank you. We've started looking and working with the radiologist at City of Hope and some animal models to do just that using MRI modality. So we're putting magnetic resonance imaging active metals in the metallocorol and starting those studies now, and it's very exciting. So I think within a couple of years, we might have the ability to start a phase 1 trial.
Wow, that's wonderful. That really is quite a thing to be able to offer people from this work. Yeah. And I know you guys have worked really hard and really in a really focused way to achieve this. So thank you for doing this work and thank you for telling us about it.
Sure. Thank you.
That was the last of our talks, and I'd like to first thank the audience for your attention. It really matters to all the scientists involved in this research that you care. I'd like to thank the scientists for the work they've done, for the hard work their laboratories have done and their collaborators. I'm sure there have been many days and nights when they had other things that they could have done, but they've been doing research instead because they know that this is important to curing cancer. I'd like to thank an unsung group of scientists, and those are the reviewers, the North American scientists from Canada and the United States, who review the applications that we receive at ICRF. The Barron Scholars Program was highly competitive, and applications were chosen from among some excellent different proposals so that we could find science that would be of the greatest kind of impact for cancer. And I'd like to remind you of the motto of ICRF, which is the answer to cancer is research, and this is what we're trying to do. And finally, of course, once again, thank you, Harvey Miller, and thank you, Bruce and Jacki Barron, for inspiring Harvey to make this amazing gift.